Now we're going to go over our study guide for our genetics test coming up on Tuesday. First one says draw a test cross. Okay, number one said draw a test cross. So this is what a Punnett square for a test cross will look like. We're going to have a recessive parent. Always have to have a recessive parent. And then we have to have a parent that's showing the dominant trait, but we don't know if that parent is homozygous or heterozygous. So that's what it would look like if we had to write it out. It would look like little n times unknown. And that would be a test cross. And next in our chart it says fill in with either homozygous dominant, recessive, or heterozygous, and purebred or hybrid. Okay, so remember hetero means different. Big R, little r, that's two different alleles, so that would be heterozygous. And heterozygous is also called hybrid. So heterozygous is hybrid. Big K, they're the same. Homo means the same. They're both big letters, so this one would be homozygous dominant. Anything homozygous is considered purebred. Little c, again, the alleles are the same, but this time they're little letters, so this one is homozygous recessive, and it would also be purebred. Number 3 says be sure you can complete and analyze monohybrid crosses using a Punnett square and show how and know the ratios and percentages. Okay, remember mono means 1, so this is when we're just looking at one trait. That's just our simple little 4 square Punnett square. And our simple dominant recessive. Remember, if we're writing as a ratio, we can write that maybe 1 to 2 to 1 or something like that. If we're writing it as a percentage, if there's 1 out of the 4, let's say homozygous dominant. If we have 1 out of 4 homozygous dominant, then that would be 25%. Count them like quarters. If it's 1, it's 25. If it's 2, it's 50. If it's all of them, it's 100%. And know the difference between genotype and phenotype. If it's asking the genotype percent, it's talking about the letters. Okay? If it's asking the phenotype percent, phenotype's a physical characteristic. So while you might have 1 big letter, and then 2 big letter, little letters, well, that would be 3 out of 4, or 75%, that would be phenotypically showing that dominant trait. Okay, so the genotype and phenotype ratios are not necessarily the same. Number 4 says, which of the following is an advantage of asexual reproduction over sexual reproduction? Okay, remember, asexual, that's like bacteria doing binary fission. They copy their DNA and just go bloop and split right in two. Doesn't take long, doesn't require an additional parent, doesn't require a lot of energy. So, which of the following is an advantage of asexual? It does not lead to genetic diversity in a population. In asexual reproduction, all of the offspring are identical, which can be detrimental to a population. It does not require specialized sex cells. No egg or sperm are involved in asexual reproduction. It does require only one parent, and it requires less energy. It says more energy, but if you don't have to go hunt down a mate and then take care of the eggs and all that, then it's not going to require as much energy. So it requires only one parent. That would be the advantage of asexual reproduction. Number 5 says, which of the following is an advantage of sexual reproduction over asexual reproduction? Well, it does require 2 parents. It does require egg and sperm cells, specialized sex cells. It does take a long time. You have to wait till you reach sexual maturity, find a mate, wait till the baby grows and all that. So it takes a long time. The big advantage, the reason organisms do sexual reproduction, the big advantage is that it leads to genetic diversity in a population, and that increases the species' chance of survival if there are changes in the environment. So it leads to genetic diversity in a population. Sexual reproduction leads to genetic diversity in a population. That is very important. There's our Punnett square. It's a heterozygous for type B. To be heterozygous, remember A and B are codominant. The recessive allele is O, so to be heterozygous B, he would be BO. A woman is type AB, so she's AB. And then our offspring would be AB, AO, BB, and BO. It says, what's the probability that the child will be type A? Well, only 1 out of our 4 is type A. Okay, so 1 out of 4 would be type A. That would be the phenotype. 1 in 4. Type A. And we could also say that that is 25%. They would have a 25% chance of having a type A child. Okay. And again, here's an example of our genotype, phenotype thing. We have 1 that is genotype BB and 1 that is genotype BO, but both of those would be phenotype type B. Okay, and this one, 25% would be phenotype AB. Number 7 says, give an example of nondisjunction. Okay, remember, nondisjunction is when the sister chromatids fail to separate appropriately during anaphase 2 of meiosis. Okay, so an example of nondisjunction, that means one of the gametes is going to end up with too many chromosomes and the other gamete is going to end up with too few. So if it happens with chromosome number 21 and that egg or sperm ends up with 2 copies of chromosome 21 and then it's fertilized, and with another chromosome 21, it has a total of 3 chromosome 21s. That would be trisomy 21 or Down syndrome. So Down syndrome would be an example of nondisjunction. Number 8 says a blank is an organism that is capable of passing a trait for a specific disease to the offspring, even though it does not have that trait itself. And that would be a heterozygous case if it's a recessive trait and the person is heterozygous. They would not show the trait, but then they would be capable of passing that trait on to their offspring. And we would refer to that as being a carrier. So that would be carrier. Carrier is someone who does not have a trait, but is capable of passing that trait on to their offspring. A carrier. And number 9, we're going to match up Mendels Laws of inheritance with their descriptions. And we have law of dominance, law of segregation, and law of independent assortment. And first one says, big F represents a gene for brown coat and little f represents a gene for white coat. In the cross of big F with little f, all of the offspring have brown coat, so they're all showing that dominant trait. They would all end up being heterozygous, genotypically heterozygous. They would all phenotypically be showing the dominant trait. So that would be A, law of dominance. The next one says, during the formation of the reproductive cells, the tall and short genes of the pea plant separate from each other. If you hear the word separate, I want you to think segregate. Remember, separate but equal, segregation. So segregate means to be separate. So they're separated, law of segregation. A random assortment of maternally and paternally derived chromosomes in meiosis results in gametes that have different combinations of these genes. That means just because you got mama's hair color gene in that egg, you might not necessarily get mama's eye color gene in that egg. You might get the daddy's eye color gene in that particular egg as they separate. That's the law of independent assortment. They assort independently of one another. You don't get all mama genes in one and all daddy genes in another. Number 10 says a test cross is a cross between a parent with a recessive trait and an unknown parent with a blank trait. But the reason that parent is unknown is because they're showing that dominant phenotype, that dominant trait. And we just don't know if they are homozygous for that trait or if they are heterozygous, because either way they would still show that dominant trait. So we have to have the first parent has to be recessive, and then the second parent would be showing that dominant trait, and we just don't know if it is homozygous or heterozygous. Number 11, in dogs, shiny coat big S is dominant over dull coat little s. If a shiny dog coat makes with a dull coat dog, and some of the offspring are shiny and some of the offspring are dull, what is the genotype of the shiny coat dog? We could do a Punnett square. Or we could remember our keywords to look for. Okay, if it says some are shiny and some are dull, some is our keyword. Some are shiny, some are dull. So if some are shiny and some are dull, then that means some, okay, the dull coat dog can only give little s's. So if some are shiny, they had to get a big S from that other parent. And if some are dull, they would have to be little s, little s. So they would have to get a little s from that parent. So that unknown parent would have to have a big S and a little s. If it says some and some, then that parent is going to be heterozygous. So this one would be big S, little s. And I would remember this question. You are quite likely to see that question again. So be very familiar with this question right here. Number 12 says, be sure you can complete and analyze blood type genetic problems using Punnett squares. Or this more genetic problems you just need. Be sure to remember when we're analyzing the blood type genetic problems that A and B are codominant. So if you have an A, it's going to totally show up. If you have a B, it's going to totally show up. And then if they're heterozygous for a blood type, then O is the recessive. Okay, so to be blood type O, they would have to be little o, little o. But if they're BO, then would be blood type B. If they're AO, then it would be blood type A. And AA, of course, would be A, BB would be B, and then A and B would be AB. So make sure you remember how to do those. I'm giving an example of codominance. And again, A and B blood types would be codominant. The alleles for A and B blood types. Also, codominant would be like if we had a white bunny and we mated that white bunny with a black bunny, and we would get a white bunny with black spots. That white is totally showing up, that black is totally showing up. So they are codominant. They are both still there, they're both still dominating, but they're dominating together. That would be an example of codominance. 14 says a sex-linked gene is located on the blank chromosome. Well, it's going to be on those sex chromosomes. So it's going to be either the X or the Y. Most of the time, we're going to see that happening on the X. Okay, so most of the time it's going to be on the X, but we will put X and Y, because those are both the sex chromosomes, so X, Y. X or Y. Okay. So the X or Y chromosomes, and the majority of what we see is actually on the X. 15 says an organism that is capable of passing a trait for a specific disease to its offspring, but which does not express the disease itself. Okay, I think we had that question worded a little bit different a minute ago. If they do not show the trait themselves, but they are able to pass it on, we call that a carrier. And remember that carrier is going to be heterozygous, so they have a dominant allele, so they are able to show that dominant trait, but they also have that recessive allele that they could pass on to an offspring. Number 16 says the abnormality in the number of chromosomes in an organism is called, I don't know if I say it's called, I would say it's the result of nondisjunction. It's the result of nondisjunction. And then it says, what phase of meiosis does this occur? It's going to occur during anaphase, and in more particular during anaphase 2, when those sister chromatids fail to separate appropriately. Number 17, red roses and white roses are cross-pollinated, producing pink roses. Okay. We had red roses, we have white roses, now we have a totally new trait of being pink roses. And pink is a blend of red and white. So what pattern of inheritance would this represent? If we get a blending, it means that red was not totally dominant, that white was not totally dominant, neither one of them showed up completely. They blended together to make a new trait. So if they blend together to make a new trait, that is called incomplete dominance. Incomplete dominance. Either one is completely dominant over the other. Number 18, a rooster has red and black feathers. This pattern of inheritance represents what? And the red and black didn't blend, make, I don't know, brown or something. The red is totally showing up. The black is totally showing up. The red is dominant in its place. The black is dominant in its place. They are being dominant together. This would be an example of codominance.